Hello! During this presentation, we will take a more in-depth look at the components needed for genetic engineering. First, let's talk briefly about source DNA. Where do scientists get their DNA? Many scientists get DNA directly from the organism. For example, bacteria, viruses, plants, humans, crime scene investigations, and of course they can also purchase many genes or even entire genomes commercially. Next we will consider plasmid DNA. Where do scientists get the plasmids? Although it is possible to design and build your own, most scientists purchase their plasmids or vectors from a wide selection of companies such as Life Technologies, formerly known as Invitrogen, Envivagen, BD Biosciences, and GenScript, just to name a few. To review, what makes a plasmid a vector? The two genes required to make a plasmid usable as a vector are an origin of replication and a gene that encodes for resistance to an antibiotic. As a bonus, most commercially produced vectors contain what is called a multiple cloning site. This is simply a short stretch of DNA that contains the recognition sequences for several restriction enzymes. This gives the scientist more flexibility when inserting their own target gene. Here is the map for a typical vector. Notice it has a gene for resistance to ampicillin, a common antibiotic, an origin of replication so that the plasmid will be reproduced when the cell splits, and a multiple cloning site, which gives the scientists 10 choices of restriction enzymes to use for their cloning experiment. So let's talk about restriction enzymes. In the early days of genetic engineering, scientists purified these enzymes themselves, but now there are a wide selection available commercially that cut at hundreds of recognition sequences. One such company that specializes in the production of restriction enzymes is New England Biolabs. As you can see, they carry a wide selection of ready-to-use restriction enzymes. One of my favorite tricks of the trade is using two restriction enzymes when building my recombinant plasmid. This way, my gene and also my vector have two different sticky ends. This ensures that the gene is inserted in the right direction. Another common trick scientists utilize is to use PCR to engineer in restriction sites where they want them. This is done by designing primers that have restriction recognition sequences followed immediately by a perfect match for the target sequence, such as adding a restriction site for EcoR1 directly before the start codon, and adding one for HIN3 immediately after the stop codon. You will notice that the stop codon is actually the complementary sequence because this is a reverse primer. When using these two primers together, the entire target gene will be produced with the added restriction sites at each end. Finally, let's look at competent bacteria. What makes them competent? Competent just means we make holes in the outer membrane and the cell wall that allow DNA to enter the cell. There are two ways to make bacteria competent. One way is to put the bacteria in a salt solution. This makes them chemically competent so that with a short heat shock, the holes form and DNA enters. Or we make electrocompetent cells by placing the cells in a weak buffer and exposing them to a brief electrical shock. Again, this just creates holes in the bacteria so that the DNA can get in. So to review, we have talked a little bit more in depth about source DNA, vectors or plasmid DNA, restriction enzymes, double digests and directional ligation, and competent bacteria. Hopefully, You now have a better understanding of how scientists actually carry out genetic engineering. Thank you for your attention.